And in the leaf structure, we have, first of all, the epidermis. And the epidermis is going to be this clear, waxy coating. Okay. It's important that the epidermis is clear because that allows light to be absorbed. It's also important that the epidermis is waxy because that actually helps us keep in the water. So our epidermis is going to be used to protect our cells, the actual cells itself, and to trap in water. Now we know how do we keep things in, but how do they actually get inside of the leaf itself? Inside the leaf you have tissue known as mesophyll, or mesophyll cells, and the mesophyll cells are where the most photosynthesis occurs, in the mesophyll, okay? So in the mesophyll cells you actually have two types. You have the palisade mesophyll and you have the spongy mesophyll. The big difference between this is that in the spongy mesophyll, this is going to have space for gases. In the palisade mesophyll, this is where most of the light-dependent reactions occur. Should make sense because it's right near the outside of the leaf, or the exterior of the leaf. But then you need this area with more gases, and so that the gases can be transferred in. Where does this occur? Or how does this occur? Through the guard cells. From Biology 1, hopefully you remember that the guard cells form a pore called a stomata. And in the stomata is where CO2 comes in and oxygen can also leave. So here you have your CO2 and O2 entering and leaving from the stomata. Now the final ingredient that you needed for photosynthesis was the water. How does the water enter the cell? Water doesn't actually enter the leaves itself, but through the roots of the plant and up the plant. So we have to have special cells or special tissues to enable that to happen. So to move water, you have tissue called xylem, and it's actually bundled up in what's known as a vascular bundle. There's also some phloem that's attached with that, or right beside that bundle, but right now we're going to focus on the xylem. The xylem is what transports water up from the roots. We'll need to know more about this later, but right now you just want to know the basics of where are you getting these reactants from. Okay? So, just as another bit of a review, what happened during our light-dependent reactions? So in our light-dependent reactions, we found that this process first starts with using light to excite electrons. Once we excited the electrons then are used to pump hydrogen ions, and eventually that electron that pumps these hydrogen ions into the thylakoid space is placed onto the carrier, electron carrier, known as NADP+, which eventually, when those electrons are actually transferred, is reduced and becomes NADPH, the electron carrier that we'll use in the next step. As the electrons are being excited and pumping these hydrogen ions through this process, and here are the hydrogen ions being pumped, okay? As the hydrogen ions are being pumped, we produce, or through chemiosmosis, we will photophosphorylate and produce from that some ATP. Although, even though we are making ATP in the first step of photosynthesis, this overall reaction of photosynthesis is going to be endergonic. Yes, we produce a little ATP, but overall, we're going to be taking or using that ATP. So, endergonic process. So again, just quick review. We're going to use the light energy at this photosystem. This is where all our pigments are. We're going to excite the electrons. As we excite the electrons will move through this chain, through the electron transport chain. As it moves through the electron transport chain, we'll go through active transport again, pump hydrogen ions into this thylakoid space. The hydrogen ions will move passively into the ATP synthase, and we will produce some ATP, as well as that electron moving through this thylakoid membrane, and finally producing this NADPH molecule, all of which go to the Calvin cycle. Another thing you just want to keep in mind is that this is the thylakoid membrane. Okay? A thylakoid membrane is literally one little stack. One little stack in the chloroplast is the thylakoid. But if you have a whole group of those membranes, then you have grana or granum. Then on the outside space, because these stacks will be found inside the chloroplast, you'll have your stroma. So the first part, the light-dependent reaction, occurs at the stroma, and now occurs at the thylakoid space or thylakoid membrane, and now we're going to move into the stroma for the Calvin cycle. Into the stroma. So, how are ATP and NADPH used to build glucose? The Calvin cycle is pretty tricky, but I'm sure you're used to that after we've seen respiration. But what we need to remember about or learn about the Calvin cycle is that we start with a molecule known as ribulose bisphosphate. We start here. And we're going to remember this molecule as RUBP for short. We need to know, and maybe this might help you figure out this process, that RUBP must be recycled. So this should be very similar to you with what we did in respiration. In respiration, we also recycled a 4-carbon molecule. But in this case, we are going to work with carbons again. And the process is going to be a little different because instead of making ATP, we're using AGPs and using electron carriers. And we're starting with, instead of a 4-carbon compound, we have a 1, 2, 3, 4, 5-carbon molecule. So, first thing, at the bottom of your paper, it asks you how many turns are needed. We're going to need 3 turns to produce 1 product. And our 1 product is going to be a 3-carbon molecule that is known as G3P. You've seen G3P before. So note to yourself that, hey, these are being recycled or used in the Calvin cycle and in respiration. So we need 3 turns. What does that mean? If you look at this picture, our input, we start with RUBP 5-carbon molecule, and notice that we actually have 3 of them in here. 3 RUBPs. And we're going to input 3 CO2s. And we're going to input these 3 CO2s simultaneously. I hope I spelled that right. Okay? So we have 3 carbon dioxides. You start with 3 of these 5-carbon molecules, and 3 of these CO2s. Each individual CO2 will simultaneously attach to the 3 5-carbon molecules. So what does that mean that you have? You end up producing an intermediate that has 6 carbons. So between here and here, you have a 6-carbon molecule that's being produced, and 3 of them. What happens in phase 1, carbon fixation, adding carbon, we create an intermediate, create a 6-carbon intermediate that is unstable, so it splits in half. So you have a 6-carbon intermediate. We have 5 carbons. Here's one more carbon. That gives us 6. However, we did this 3 times. So you have technically 3 6-carbon intermediates. Then what happens is they split in half and produce 2 of these each. So notice here you have a 3-phosphoglycerate molecule. How do you have 6 of them? Well, you actually had 3 of these 6-carbon intermediates. They had 6 carbons each. We split them into halves. And so now we have 6 of the 3 carbons. If you add up your carbons, it should make sense. 5 plus 1 equals 6. 6 times 3 that it's happening gives you 18. 18 means I have 1, 2, 3 in one of these, and so 3 times 6 gives me my 18 carbons. Then what happens next? Well, then what we're going to do is we're going to add on ATPs, we're actually going to add on phosphates from ATP, technically this one, and we're going to add on electrons from the NADPH. So reduction, you just add electrons. Then what happens is you produce glycerol aldehyde 3-phosphate, G3P, which we've seen before. We produce 6 of them, but because we need to recycle this molecule, we're going to continue moving these 5 extras along and produce only 1 G3P molecule. Okay? So what you want to do is just think about, well, what's happening to the carbons? I have 6 G3Ps, but I'm only going to let 1 leave this process. Because only 1 leaves this process, the others can actually be recycled. And those that are being recycled will be regenerated into RUBP. We will talk about this some more because I know it's tricky. But what do we actually use during the Calvin cycle? In the Calvin cycle, we use, or total use, is 9 ATPs and 6 NADPHs and 3 carbon dioxides. And all we end up making or producing is 1 G3P. This had to happen 3 times. We had to use 3 carbon dioxides, and we only produce 1 G3P. If we add on another G3P, we can make glucose. If we can add on a bunch more, we can make starch or cellulose. But we're going to stop there today.